Hi everybody, this is Mrs. McQuaid, and today we're going to be talking about science as a way of thinking, questioning, gathering evidence, and learning about the natural world. In other words, the scientific method. If you'd like to print out slides to follow along or take notes on, you can visit my website at www.sevengablesscience.com and select Biology, Semester 1, Unit 1. Let's get started. The objectives for this section are as follows. Students will be able to identify the different elements of scientific inquiry, create testable hypotheses, differentiate between independent and dependent variables, and explain the importance of a control group. Important vocab to keep an eye out for include the following words: science, hypothesis, observation, control group, constants, which, depending on which textbook you're using, are also known as controlled variables, independent variable, dependent variable, and data. These are words you'll be responsible for on the end of unit assessment. So what is science? Throughout this course, you're going to learn a lot of facts and ideas, and while they're important, it would be a mistake to think of science as just a collection of never-changing beliefs. Scientific ideas are open to testing and discussion and even revision. This is because science is a way of knowing. It's an organized way of gathering and analyzing evidence about the natural world. And by that, I mean parts of the world that we can measure and manipulate. Scientists collect and organize information in an orderly way, looking for patterns and connections, and they propose explanations based on evidence, not belief. I want you to think of science as a verb because it's something that we do. It's really a way of problem solving. The goals of science are to provide natural explanations for events in the natural world, and to use those explanations to understand patterns in nature and make useful predictions about natural events. While the scientific method can look daunting, it's really just a method of problem solving, which is something that people do all the time very naturally. The difference is that a scientist approaches research in a very systematic way, developing and testing hypotheses through experimentation. They ask questions, they do research, they construct a hypothesis which they can test, and then they analyze data to draw their conclusions. Science doesn't really end with the end of an experiment. Oftentimes, the conclusions that we draw lead to more questions, which we can then go back and research. Scientific investigations usually begin with observations, which are the act of noticing and describing events or processes in a careful, orderly way. Scientific observation involves more than just looking at something. It's looking at something that everybody has seen and thinking something that nobody else has thought yet. It's the kind of observation that leads to asking questions that haven't been asked before. For example, at some point in your life, you've probably noticed that after it rains, there are earthworms everywhere. They're all over the roads and sidewalks, and maybe you've wondered why. That's the beginning of scientific thinking. After making observations and figuring out which questions you want to answer, a scientist will make inferences, which are logical interpretations based on what the scientist already knows. Inferences and creative imagination lead to the formation of a hypothesis. A hypothesis is a scientific explanation for a set of observations that can be tested in ways that support or reject it. For an experiment to give valid results, it must be controlled. To design a controlled experiment, you must have two groups, an experimental group and a control group. Your experimental group is the group that you're going to be testing. Your control group is the group that you use to compare your experimental group to. This is how you're going to see if the thing that you're testing causes a change. In other words, if you should support or reject your hypothesis. For example, consider brownies. If you've ever made prepackaged brownies before, you'll know that the box calls for vegetable oil. But my mom thinks that butter makes everything better, especially in brownies. So she replaces the vegetable oil with butter. Now, if you wanted to know if it really makes a better brownie, you can't just make one pan of brownies. You need to make two. A pan of brownies with butter and a pan of brownies with oil. That way you can try both brownies and see which one is actually the better brownie. I'm not going to tell you which makes the better brownie, but I strongly encourage you to do the experiment and find out for yourself. Okay, so we know we need two groups in an experiment, our experimental group and our control group, to design a controlled experiment, but we need to take a closer look at them. Experimental studies allow scientists to determine what causes a particular phenomenon, and in experiments, there are three types of variables that scientists will focus on. Independent variables are the things that you're manipulating or purposely changing. Dependent variables are the things that you're going to observe or measure at the end of the experiment. Independent variables affect the dependent variable. So going back to my brownie example, the thing that you were testing or curious about was the butter, and the thing that you're measuring was the taste. So that makes the independent variable butter and the dependent variable taste. Experiments also have controlled variables. Those are all of the things between your experimental group and your control group that are going to be kept the same. So for example, in our brownie case, it wouldn't be right to use Ghirardelli brownie mix for one of your trays and Hershey's brownie mix for a different tray. It also wouldn't be right to use 2 eggs in one of your brownies and 1 egg in the other tray. You have to keep all of those things the same in order to know that the taste of the brownies being different is due only to the butter being used. Another thing that I want to point out, because oftentimes in science, data is represented in a graph. Whenever you are graphing scientific data, you're always going to put your independent variable down here on your x-axis and your dependent variable on your y-axis. So if you are asked to identify the independent and dependent variable in an experiment and there's a graph, it's usually pretty easy to do because you just have to read the titles of the x and the y axis to figure it out. So let's go ahead and practice that. Go ahead and pause the video and identify in a notebook your independent variable and how you know it's your independent variable, your dependent variable and how you know, and see if you can write a sentence identifying the relationship between your independent and dependent variable based on the data in the graph. Did you do it? Okay, so let's check. The independent variable is going to be time, and you know this because you can see it down here on your x-axis, and your x-axis is always going to have your independent variable. The dependent variable is going to be population size, and you know that because it's found on the y-axis. Now, when you see a question asking for the relationship between your independent variable and your dependent variable, what that means here is what's the relationship between time and population size. So you could say something like, as time progresses, the population increases. All right, let's try another one. So again, go ahead and pause and write down your answers identifying the independent variable and how you know, the dependent variable and how you know, and see if you can describe the relationship between the independent and dependent variable. All right, so let's take a look at this graph. It's a little bit different than the last one we looked at. This is called a scatter plot because you see all these dots, and then through the middle of the dots, you have kind of this red line that's been drawn, and this is called a line of best fit. And it kind of just shows you the trend of the data. So looking at this graph, we would say that the independent variable is precipitation in millimeters, and we know that again because it's on your x-axis. Your dependent variable is cucumber yield, and we know that because it's found on the y-axis. And when asked to describe the relationship between precipitation and cucumber yield, you would say that the more precipitation you have, because you could see that the numbers are getting higher here, so the more precipitation you have, the higher your cucumber yield. And that makes sense, right? Because if a cucumber plant gets more rain, it's probably going to have more materials to grow more cucumbers. All right, so those were a couple easy things. Let's go ahead and revisit a hypothesis, because a hypothesis, writing a good one can be tricky. So hopefully you're feeling a little more comfortable with identifying independent and dependent variables, because that's an important step to writing a good hypothesis. The first thing we want you to do whenever you write your hypothesis is remember that it's a statement, not a question. Your hypothesis is not the scientific question in your project, and this can be confusing because oftentimes in elementary school we're taught that your hypothesis is a question, but you're a little bit older now and the subject is a little more complex, so we want to clarify that it's never going to be a question. It's always going to be a statement, an educated, testable prediction about what you believe will happen. You want to make your hypothesis clear. A good hypothesis is written in clear, simple language. Reading your hypothesis should tell your audience what you thought was going to happen when you started your project. It's okay if you end up being wrong, but it still needs to be a predictive statement. A good hypothesis is going to be clear and understandable. It'll explain what you think will happen. And very importantly, it's going to identify those independent and dependent variables. So you want to clearly state what you're testing, that's your independent variable, and what you predict the effect of the changes will be on what you're measuring, your dependent variable. Finally, if you didn't already know this, a good hypothesis has to be testable. We have to be able to either prove or disprove your hypothesis through an experiment. If you can't do that, it's not something in the realm of science, and it's not a good hypothesis. So now that we've looked at the components of a good hypothesis, let's go ahead and practice picking the better hypothesis. So let's look at pair number 1. A says, when there's less oxygen in the water, rainbow trout suffer more lice. B says, do rainbow trout have more lice when there's more or less oxygen in the water? Hopefully you've chosen A as the better hypothesis. Remember that a hypothesis is not a question, it's a prediction. So you can see that in part B, what we have is a question, so that's not going to be right. In part A, you have a statement. It identifies your independent variable as the amount of oxygen in the water. It identifies your dependent variable as the number of lice. And it's predictive because it says if there's less oxygen, then there will be more lice. So A is going to be your better choice. Hypothesis pair number 2. Our universe is surrounded by another larger universe with which we can have absolutely no contact. Or the more high temperature gases in galaxies, the more X-rays they will emit. Hopefully you've chosen option B. Remember that a hypothesis must be testable. If we can have absolutely no contact with something, you definitely can't manipulate and test it. In B, we can see that the independent variable is the higher temperature, so that's identified. We can see that the thing we're going to measure, the dependent variable, is more X-rays, and there's a predictive element there. So B is your better hypothesis. Okay, one more like this. Aphid-infected plants that are exposed to ladybugs will have fewer aphids after a week than aphid-infected plants which are left untreated. Or, ladybugs are a good natural pesticide for treating aphid-infected plants. Now, if you don't know what an aphid is, it can be kind of easy to get hung up on that word. So I've included a picture here. Aphids are these, sorry about that, are these tiny little green bugs that you see on this plant. And obviously this big guy here is your ladybug. So what they're saying is that in part A, it says aphid-infected plants are exposed to ladybugs will have fewer aphids after a week than when left untreated. In other words, when left without ladybugs. Part B says ladybugs are a good natural pesticide for treating aphid-infected plants. These can be a little bit tricky because they both kind of get at the independent and dependent variable. So the independent variable, the thing you're testing, is the presence of ladybugs, and your dependent variable is going to be your number of aphids. But Part A is giving you a better option, and that's because it is more clear and it's predictive, right? So more clear in that it gives you a definitive amount of time. After a week, and it tells you what you're going to see, there will be fewer aphids compared to plants that haven't been treated with ladybugs. So A is really going to be your better option. All right, so let's try putting all of these parts of an experiment pieces together. So here you can see we have a data table. It says seedling 1, 2, 3, 4, and 5. Soil, so we have yard dirt, sand, clay, Miracle-Gro potting soil, or none, and then you have a description of the different soil types. And then in the far right column, you have plant height at 10 days, and you can see that they've been measured in inches. So in this experiment outlined, 5 identical tomato seedlings are planted in 5 identical pots, placed in the same location, and watered on identical schedules. Pause the video and see if you can identify your independent variable, your dependent variable, the controlled variables, and try writing a hypothesis for this experiment. Did you try it? Okay. You should have identified the independent variable as the type of soil, the dependent variable as the plant height at 10 days. Your controlled variables, those are all the things that are identical between your different seedlings, are going to be the identical pots placed in the same location and watered on identical schedules. And for writing a hypothesis, you might say something like, in this experiment, plants grown in Miracle-Gro potting soil will grow taller after 10 days than plants grown in other types of soil. If you said that, then based on these results, you would probably say that your hypothesis would be supported, right? Because you could see that the Miracle-Gro potting soil plant grew 10 inches, which is taller than all the other options. But I just want you to know that you would have also been correct if you wrote a hypothesis that said plants grown in clay soil will grow taller after 10 days than plants grown in other soil types. Remember, we don't have the results of a hypothesis before we run the experiment, and sometimes our hypothesis is wrong, but as long as you've clearly identified your independent variable and your dependent variable, what you're going to measure, your hypothesis would be marked correct. If, however, you had said clay was going to help the plants grow taller, and obviously we can see that the plant grown in clay only grew 2 inches, which isn't as good as the yard dirt or the Miracle-Gro. At the end of the experiment, you would just say, my hypothesis was rejected, and that's okay. For more resources, including printable notes, worksheets, and access to online practice, visit my website at www.sevengablesscience.com and like and subscribe to my YouTube channel, Seven Gables Science, to keep learning.